Hello, and welcome to Virtual Investor Conferences. My name is Matt Lateplo, and on behalf of OTC Markets, as well as our co-host, Zach Small Cap Research, we're very pleased you have joined us for our next live presentation from Lisata Therapeutics, Inc. Please note you can submit questions for the presenter in the box to the left of the slides. You can also view a company's availability for one-on-one meetings through the Schedule Meetings tab found on the conference platform. At this point, I'm very pleased to welcome the next presenters. David Mazzo, President and CEO of Lisata Therapeutics, Inc., which trades on Nasdaq under the symbol LSTA. Welcome back, David.
Thank you, Matt, and thank you, everyone. I truly appreciate you taking the time to learn about Lisata and to hear about the progress we've made since we've last had an opportunity to speak to your audience. So Lisata is a public company, and I will remind everyone that we will be making forward-looking statements during the presentation, and I ask you to keep those in mind as you make investment decisions. So let's start with a quick company overview. What is Lisata? Well, Lisata is a clinical stage therapeutics development company working in the area of cancer in oncology, and we're working on a novel solid tumor targeting and penetration technology that's designed to improve the efficacy of anti-cancer agents, which are co-administered with our products. Like many biopharmaceutical companies, we have a seasoned management team with successful international drug development experience and expertise. Many of us have worked in both big pharma and small pharma and have developed drugs around the world successfully. Of course, our technology has intellectual property protection, and as you'll hear later, we also have market exclusivity protection based on orphan designations. But most importantly, we have multiple projected product and business development milestones over the next 12 to 24 months. All of those should lead to value creation for our investors. We also have a platform technology that we would indicate as being validated by the existence of a number of partnerships that we've created with other companies that I think gives a sense of the potential for the technology that we're developing. And maybe most important of all in this era of difficult financing for small cap companies, we have 2 years of cash on our balance sheet. Our projected cash runway goes into early 2026, and we have funding for all of the development programs through data that you'll hear about today. So let me tell you a little bit more about what we're doing and why we're doing it. So of course, we're working in cancer, but more specifically, we're working in solid tumor cancer treatment. That remains a global vital need because more than 90% of the newly diagnosed cancer cases are solid tumor cancers. And these are the cancers that people dread being diagnosed with, cancers like lung, breast, pancreas, liver, etc., which have very poor prognoses in today, even with today's advanced medicines. The reason that solid tumor treatments remain suboptimal is that targeting and penetrating tumors present distinct challenges biologically. And that's because these tumors are surrounded by a mixture of cells which contain tumor cells, tumor endothelial cells, fibroblasts, etc., which constitutes something called the stroma. And this stroma encapsulates these solid tumors and acts as a physical barrier, limiting the penetration and distribution of anti-cancer agents into the tumor. In addition, these tumors present a microenvironment of immunosuppressive cells, which contributes to tumor resistance to drugs, as well as metastases or the spreading of the cancer to other organs throughout the body. And simply prolonging or escalating the dosing of non-targeted anti-cancer therapy doesn't solve the problem because it generally leads to intolerable off-target side effects. So we have decided that there is a way to address both targeting and penetration by harnessing something called the C-end rule, which is a biological active transport mechanism which exists naturally and in many cases is used by cells to transport nutrients into the cell. We hijack that to transport anti-cancer agents into these tumors. And you start with the concept that something called RGD peptides are very well known and very efficacious tumor targeting agents, but they do not enhance penetration and delivery. One of our founders, a professor by the name of Erkki Ruoslahti, developed something called the internalizing RGD peptide, which combines targeting and penetration enhancement. And our lead program, LSTA1 or certepetide, is an internalizing RGD that triggers the C-end active transport mechanism, for selective enhancement of intratumoral delivery of anti-cancer drugs. And LSTA1 is now in mid to late stage clinical development as a solid tumor treatment. It promises to convert tumor stroma from a barrier to a conduit for anti-cancer drugs. And importantly, it also combats resistance and metastases because it's been shown in preclinical experiments that LSTA1 selectively depletes immunosuppressive T cells in the microenvironment, enhances the cytotoxic T cells, and inhibits the metastatic cascade. And what's also important is that LSTA1 is agnostic from a mechanism of action perspective to the modality of the companion anti-cancer drugs. We can combine it with chemotherapy, immunotherapy, radiotherapy, antisense or sRNA therapies, etc. And the mechanism of action remains both selective and tumor penetration enhancing. So based on this, we're developing the product in a two-pillar strategy approach. On the one hand, we're pursuing rapid global registration in pancreatic ductal adenocarcinoma, or MPDAC, initially combined with gemcitabine nab-paclitaxel standard of care. Gemcitabine nab-paclitaxel is dosed to approximately 50% of the people with pancreatic cancer. The other standard of care is FOLFIRINOX, and the other 50% receive that. We have actually completed enrollment in a large phase 2b trial in pancreatic cancer, and we'll be expecting to report top-line data from that trial later this year. In parallel, we're working to demonstrate that LSTA1 effectiveness is evident when we combine it with a variety of standard of care regimens, including, as I said, chemotherapy, immunotherapy, etc., in a variety of different solid tumor cancers. And here we have multiple phase 1b/2a proof of concept studies ongoing throughout the world. So let me mention also a few of the partnerships that have been in place, because it's key here, because through these partnerships, as well as selective geographical placement of our studies, we're able to manage our capital very, very efficiently. And so we've got a number of development alliances, including with a testing group in Australia and New Zealand called the Australasian Gastrointestinal Testing Group, as well as with a foundation, a philanthropic group called WARP9. And these groups contribute resources, people, money, etc., but they take no commercial interest in our products. And so this is one way we keep our costs low and maintain control over the commercial viability of our programs. In addition, we do have one strategic commercial partnership with a large pharmaceutical company in China called Qilu Pharmaceutical. And Qilu is moving into phase 2 in development, working under a joint steering committee with us for Chinese registration of that product, with a typical biobucks type of deal, with still approximately $200 million of milestones and royalties available to us as they proceed through development. And we have a number of additional opportunities that exist for many combinations with many different companies or very many different tumor types, because, as I said, we can combine LSTA1 with almost any modality of anti-cancer agent. So let me explain how this works. It's actually quite elegant, and this mechanism of action and this approach was actually the basis for Erkki Ruoslahti's Lasker Award, which he received last year. Many people know the Lasker Award as probably the precursor to a Nobel Prize in medicine or chemistry in many cases. So this has been already recognized as a major breakthrough in biochemical research. So LSTA1 is a 9 amino acid cyclic peptide, and it has a very high binding selectivity and affinity for alpha V, beta 3, beta 5 integrins, which are significantly upregulated on the cells that comprise the tumor stroma. And once LSTA1 is bound to these integrins, it becomes susceptible to protease cleavage due to proteolytic enzymes, which are naturally occurring in the tumor microenvironment. And when the cleavage takes place, LSTA1 then is broken into 2 linear peptides, one of which, the 5 amino acid fragment, is a C-end rule fragment, which has a very high binding selectivity and affinity for an adjacent receptor called neuropilin-1. And when neuropilin-1 is activated, it actually actuates the C-end rule transport mechanism. And that's manifested by the formation of microvesicles at the surface of the stromal cells, which encapsulate those anti-cancer drugs in the circulatory system and ferry them or percolate them through the stroma to deposit their payload deeper into the tumors. In addition, when the C-end rule is activated, the gap junctions between the cells of the stroma relax or open, and that allows for greater infiltration of those cytotoxic T cells, those immune cells that we talked about earlier, to also contribute to the combating of the cancer. We have a number of preclinical work studies that demonstrate the high degree of selectivity and penetration enhancement. In this particular model, we have a nude mouse which has been implanted with a human pancreatic ductal adenocarcinoma tumor. It's where the arrow is shown. And then we dose the mouse with something called fluorescent quantum dots. When those FQDs are in circulation, if the mouse is asked to fluoresce, if it's excited, you would see the entire mouse turn green or fluoresce. When we quench that with a quenching solution, any of the FQDs in circulation go dark. And you can see in the first panel where only FQDs and etching solution are applied, the entire mouse is dark, which means there was no selectivity to the distribution and penetration of the fluorescent quantum dots. On the right, however, if we co-administer LSTA1 with the quantum dots and then go through the etching experiment, you can see all of the fluorescence is now sequestered in that particular tumor, showing the high degree of tumor targeting and tumor penetration. And that's complemented by approximately 300, and now we're up to 358 scientific publications in peer-reviewed journals from around the world, which demonstrate that when you combine LSTA1 or LSTA1 analogs, other IRGDs, you see a consistent improvement in the efficacy of the co-administered anti-cancer agents, regardless of their type, regardless of the validated solid tumor model. So very compelling preclinical data, but really what's most important is what happens in people. And here are the results from our phase 1b/2a trial done in Australia several years ago. This was in first-line MPDAC patients, 3 sites throughout Australia. Patients were dosed with LSTA1 in combination with gemcitabine, nab-paclitaxel. And being a phase 1 trial, one of the key issues was safety, and we were very pleased to report that LSTA1 was extremely well tolerated. There was no dose-limiting toxicity that could be identified, and the safety of LSTA1 in combination with the chemotherapy standard of care was consistent with the safety of the standard of care alone, meaning that LSTA1 did not exacerbate side effects. But what it did do was significantly augment the efficacy of the co-administered chemotherapy, as you can see on the right-hand side, where there is significant increase in all of the cytotoxic endpoints. But I think it's easier to see when we look at this graphically. There was a 55% improvement in median overall survival and a 76% improvement in median progression-free survival. Our principal investigators here in Australia characterized this as breakthrough type of data, unprecedented improvement in these highly, highly sick patients who often have only 6 to 9 months to live upon diagnosis. What's also critical is what happens to all of the secondary and tertiary endpoints in a clinical trial, any clinical trial, but especially here. You want to see all of the endpoints moving in the same direction, that is consistency of clinical results. And here you can see in every case, the endpoints improve notably when LSTA1 is co-administered with the standard of care. So these very positive results led us to think about moving into a broader phase 2b program, which I'll describe in a moment. But we also had concomitantly some very promising results in other tumor types. And here is a case study that was published just a few months ago from a patient that received LSTA1 when being treated for metastatic gastroesophageal adenocarcinoma, another very deadly and difficult to treat solid tumor type. And here we had a patient, you can see the details of the patient on the left side of the slide, who had been treated with standard of care FOLFIRINOX in combination with immunotherapy, that's pembrolizumab. and after a number of courses of treatment, really only was receiving a partial response. It was not very positive. After cycle 7, we provided LSTA1 as a compassionate use opportunity here for this patient, and they combined LSTA1 with the chemo and immunotherapy standard of care. And after a number of additional combined therapy courses of treatment, and that is, you can see here after cycle 18, the patient underwent exploratory laparoscopy, and to the astonishment of the treating oncologist, there was no disease present. So this patient had a complete remission in a very, very difficult to treat cancer. Recognizing this is simply an N of 1, but still it shows the great promise and the versatility of LSTA1 in combination with, again, almost anything treating solid tumor types. And so this has now led us into a very broad portfolio that I briefly described earlier. And I want to talk about the fact that we have received regulatory accolades which corroborate our sense of the promise of LSTA1 to be able to make significant improvements in patient treatments in these deadly diseases. We've already received Fast Track designation in the United States for pancreatic cancer. And we've also received orphan drug designation in the United States and Europe for pancreatic cancer, and in the United States for malignant glioma or brain cancer. And as you can see, described here on the slides, for those of you who don't have the details, these are more than just regulatory badges of honor. They provide us with monetary and commercial benefits that allow us to get through development quicker, get through review processes at the regulatory agencies more quickly, and in the case of orphan drug, actually extend market exclusivity. So these are very important from a value-driving perspective as well as a medical and regulatory perspective. So the next two slides just are a tabular listing of all of the trials that we have ongoing. As we said, we are funded to complete all these trials through data, and that's again because of the fact that we have partners who are contributing or in some cases entirely funding the trials, and we are also doing the trials in geographies where we receive R&D rebates or where the costs are generally lower, but where the clinical quality remains very, very high. And you can see that the top line here, the ASCEND trial, 158 patients, double-blind, placebo-controlled, randomized, phase 2b trial being done in Australia, New Zealand, where we have now completed enrollment. It was announced last December, and we're in patient follow-up with data expected in the fourth quarter of this year. That will be seminal for the company and probably will be the opportunity to be a catalyst for a number of potential partnerships that could come to fruition. In addition, we have the BOLSTER trial, which is a trial that's working in cholangiocarcinoma as well as head and neck cancer, again, two very difficult to treat cancers. And that BOLSTER trial is being run internationally and is enrolling quite well. And then a number of other trials that are moving along toward completion, including trials in glioblastoma, peritoneal carcinomatosis, and others. And then, of course, we've listed the trials here of our partner in China, Qilu, who is expected to start their phase 2 trial in the coming quarter. When you map that out over the course of the next 2 years in terms of execution and data milestones, you can see there's a very rich news flow environment, again, with the phase 2b data from ASCEND, top line being reported in the fourth quarter of this year, and then data opportunities and news flow opportunities from all of the other studies throughout the year and all the way out into the end of 25 and into early 2026. All of these supportive and helpfully moving us forward toward a registration. I will mention that we've already had discussions with the regulatory authorities in Australia relative to ASCEND, and we've been told that if the data are positive from that trial, then they would accept an accelerated approval application in Australia for that product, which could lead to commercialization under ideal conditions in Australia as early as 2026. And we will follow up those conversations with similar discussions with the FDA and the EMA once data is present, but clearly ASCEND will allow us to create the most efficient single phase 3 trial that would be either necessary for registration or commitment for post-registration if we were to get accelerated approval. So ASCEND is a very important milestone for us. As I mentioned, we have cash through early 2026. We just reported our full year results from 2023 just last week. The 10K has been filed. We reported over $50 million in cash on our balance sheet, still no debt, and a very clean cap table going forward. And so I will summarize here by simply saying that we believe that we represent, if I can use the term, unicorn today in the small cap biopharmaceutical world. We are a company that is well financed. We can run all of our programs through data. We have a lot of news flow projected, both in terms of execution, data, and most likely business development as well. Partnerships that are already in place and others that could be formed, and a very experienced team that has been very good stewards of capital and has now developed a strong reputation and credibility for executing what we say we're going to do when we say we're going to do it. So strong people, strong technology, milestones, a solid financial situation, and partners, I think, lead to a really strong opportunity to make Lisata an attractive investment opportunity. And we hope that those of you who are listening today will consider making an investment in our company. We also have an ATM open through H.C. Wainwright, so we can do an at-the-market deal at any point in time, as well as we have the flexibility to do PIPE investments for those people who would like to do something outside of the ATM. So I thank you very much for your attention, and I now have the opportunity in a few minutes here to answer some questions. All right, so my first question here is, do we have any updates from the Ascend phase 2 trial for the first treated patients? And the answer is yes and no. As I mentioned, the Ascend trial is double-blinded, so we cannot really speak to whether or not the patients who are having a response or not having a response have been treated. But so far, the data would indicate that there should be a treatment effect. That is, there seem to be less events in the trial than one would expect historically from just standard of care. So we remain very optimistic that we will see we will see a result from the treatment of these patients who did receive LSTA1. How does one find out more about your trials in the US for glioblastoma? So the trial for glioblastoma is actually running right now in Latvia and in Estonia. It was done there for a couple of reasons. The biggest one is that for some reason in the Nordic countries, there's a higher prevalence of brain cancer than in many other parts of the world. And so it was a higher medical need and easier to recruit in those patients. Of course, if someone was willing to go to those countries and receive treatment there, they could be able to do that. But we do not have any glioblastoma trials running in the United States at this point in time. Following a successful trial in Estonia and Latvia, we would likely move to further clinical development here in the United States. And then we're asking what is our shareholder makeup and how did we raise $50 million? Well, in fact, we raised quite a substantial sum of money during the COVID pandemic. We were developing a number of other products at that time. And also we had done that raise advertising the fact that we were actively looking to broaden our portfolio and to bring in what could be a breakthrough technology, in which case is what we did when we brought in the LSTA1 technology, which was through an acquisition that we made. Since that time, which is in 2022, we have not done a raise. And the shareholders who participated in that raise made up a group of people who were both shareholders of what was our predecessor company prior to the acquisition called Caladrius, which is a number of research foundations and healthcare investment funds, as well as high net worth individuals, family offices, and a number of retail investors as well. Okay. Can you provide some more color on how you can raise additional capital via data milestones? Well, I think, there are really several ways that we can do that. The first would be that, it's not atypical for fundamental investors in the healthcare space to see an opportunity as a company has a product approaching a big data milestone and to buy into that company's stock early in order to get a better price, if you will, take on a certain amount of risk, but to actually maximize the possibility of a very large reward based on positive data. So that's a possibility. The other possibility is something similar on the business development side, where you have companies who are looking to create a license with us for LSTA in combination with their products or in certain tumor types or even certain classes of anti-cancer drugs. They could do that prior to data, perhaps to try to get a better deal, or immediately post data. And those types of deals all include generally generous upfront payments, which would contribute significantly to our cash balance sheet. And then once data is out, we could do a standard equity raise in the market, which would generally be done at a much higher price than today's prices, given that the market would generally recognize the potential of the product once positive data is out. So all those things remain possibilities for additional capital raises over the course of the next several years. And I think those are all the questions that I see at the moment. Are there any other questions out there or any that I'm missing? All right. I don't see any further questions on my screen. So if we have no further questions, I will thank you all again for listening. You can reach out to us at lisata.com or through our corporate communications Vice President John Mendito, whose contact information is on the slides. Also, if you go to our website, you can receive a copy of the slides, and you will see in the appendix there details of each of our clinical trials.